Today, we're going to be focusing on a tool that is essential to scientists worldwide, Polyacrylamide Gel Electrophoresis. Polyacrylamide Gel Electrophoresis, or PAGE, is a powerful tool used to analyze DNA, RNA, and protein samples. When polyacrylamide gel is denatured after electrophoresis, it provides information on the sample composition of the species. There are two types of PAGE that we typically talk about, SDS PAGE and native PAGE. Now you might be wondering, how does the magic of PAGE happen? Well, first, it is important to note that a positive and negative electrode are involved in all forms of PAGE. Additionally, PAGE also involves wells within the gel, where proteins are added, typically on the side of the negative electrode. In SDS PAGE, the gel is cast in a buffer containing sodium dodecyl sulfate, or SDS, an anionic detergent. SDS denatures proteins by wrapping around the polypeptide backbone. Because SDS exposes the negative backbone of the sample will primarily separate by mass, with smaller samples migrating further away from the negative electrode than larger ones. In native PAGE, proteins are separated according to the net charge, size, and shape of their native structure. Electrophoretic migration occurs because most proteins carry a negative charge in alkaline running buffers. The higher the negative charge density, the faster a protein will migrate. So, how do we analyze this information? Once the fragments have been separated, we can examine the gel and see what sizes of bands are found on it. When a gel is stained with a DNA binding dye and placed under UV light, the DNA fragments will glow, allowing us to see the DNA present at different locations along the length of the gel. A well-defined line of DNA on a gel is called a band. Each band contains a large number of DNA fragments of the same size that all travel together down the gel. By comparing the bands in a sample to the DNA ladder, we can determine the approximate size of these parts and sort them out by their composition. So, why is PAGE important and why do we need it? Polyacrylamide gels have three major advantages over Argos gels. Firstly, their resolving power is so great that they can separate molecules of DNA whose lengths differ by as little as 0.1%. They can also accommodate much larger quantities of DNA than Argos gels. In fact, up to 10 micrograms of DNA can be applied to a single slot of a typical polyacrylamide gel without significant loss of resolution. Lastly, DNA recovered from polyacrylamide gels is extremely pure and can be used for the most demanding purposes, such as the microinjection of mouse embryos. So now that you know more about PAGE and why it is important, you might be asking yourself, what are some common errors that can appear when carrying out PAGE, and what are the ways that we can troubleshoot these errors? One major error that you may encounter while conducting PAGE is the presence of distorted and smeared bands, or just bands that appear abnormal on the gel. One cause may be a high salt concentration. Salt is usually used in gel electrophoresis as it can improve or increase the conductivity of the solution, and in turn enhance the migration of proteins to the other end. However, too high of a concentration can smear or distort the bands. In order to troubleshoot this, one method involves precipitating the protein samples using TCA, a type of acid. This can concentrate the protein sample and remove the excess salt contaminants. Another cause may be overloading protein within the walls of the polyacrylamide gel. Overloading wells can lead to distorted, smear bands, as well as potentially poor band resolution and sample leakage. To troubleshoot this, a general rule of thumb is to fill no more than 3/4 of the capacity of a well. Additionally, depending on the size of the well and the thickness of the gel, between 0.5 to 4.0 micrograms of purified protein should be loaded. This error can also occur due to running the gel at too high of a voltage, resulting in the gel warming up too quickly. To troubleshoot this, a common practice is to run the gel at 10 to 15 volts per centimeter. Applying voltage is also dependent on how large the gel is and how long the runtime is. Generally, the larger the gel, the higher the voltage you should be using, and the longer the runtime, the lower voltage that should be used. This error can be caused by the undissolved material within the SDS sample buffer as well. Some proteins, such as histones, may not completely dissolve in the buffer after heating. If not removed, these undissolved insoluble materials will cause streaking within the polyacrylamide gel. To troubleshoot this, insoluble material can be removed from the sample by centrifugation before loading into the wells of the gel. Lastly, band smearing can also be caused by leaching or contamination of chemicals from the plasticware. In this case, certain chemicals from the lubricating agents used in a molding process of the gel preparation, such as olamide, can disfigure or disrupt the bands. To troubleshoot this, it requires washing out the plastic material containing the gel with either methanol or DMSO before running the gel electrophoresis process. Another major error that can occur while the conduction of page is happening is the loss of bands. A potential cause is underloading the gel wells. Again, the typical range of loaded protein should be anywhere between 0.5 and 4 micrograms depending on the gel size. To troubleshoot this, it is important to first determine the concentration of protein sample before loading. This will allow you to adjust the concentration to an appropriate amount within the recommended range for the page procedure. Another potential cause of missing bands is that the protein samples were not fixed in the gel. Generally, the process of protein fixation involves denaturing and precipitating the protein into insoluble components that then allows it to fixate onto the gel. If there is no fixation, the bands will be lost. Troubleshooting this can involve using a chemical mixture that can help facilitate the protein fixation process. One way to do this is to soak the protein gels in 45% methanol, 45% water, and 10% acetic for an hour. Alternatively, for a more stable fixative solution that can be stored for a longer period of time, the gel can also be soaked in a mixture containing 25% isopropanol, 65% water, and 10% acetic. The last major error for conducting page we'll discuss is poor band separation within the gel. This could be caused by polyacrylamide gel that is not completely polymerized, which usually results from polymerizing the gel at incorrect temperatures or for too little time. This can be troubleshooted by allowing the gel to polymerize at room temperature and to save a bit of the gel solution used to make the gel block. When the gel solution has polymerized, the gel would also have polymerized between the plates used to form the gel block that can be used in page. This error can also be caused by high concentration of acrylamide and uneven gel casting. This can result in decreasing the protein band resolution. To troubleshoot this, the acrylamide percentage in the gel can be reduced and properly cast the gel. Overall, conducting page can be a challenging task, with many different errors that can potentially occur. Being on the lookout for these important points can not only save you a lot of time in the lab, but also ensure more accurate results. Thank you all so much for watching, and thanks to the Demystifying Research course for this opportunity.